Hi everyone, my name is Lorena Alvarez and I am a part of Dr. Benjamin Greenwood's lab, and today my poster presentation is going to be determining whether dopamine receptor signaling mimics the effects of exercise in enabling stress resistance. So for some background, it is known that depression and anxiety are two of the most common mental health disorders in the world and are often precipitated by stressful life events. In rodents, behaviors similar to depression and anxiety are observed following exposure to inescapable stress, also denoted as IS, and an example of this type of behavior includes social avoidance. Now, it's been found that chronic exercise in the form of wheel running protects rats from behaviors induced by IS, similar to how exercise can protect humans against stress-related disorders. We've also found that wheel running prevents the anxiety-like effects of IS by constraining the activity of the dorsal raphe nucleus, but the mechanisms by which exercise constrains the dorsal raphe nucleus is unknown. And wheel running has also been seen to increase dopamine release and the activity of dopamine D1 receptor expressing neurons in the dorsolateral striatum during stress. So now the goal of this experiment is to determine whether increasing dopamine D1 receptor signaling in the dorsolateral striatum during inescapable stress can mimic the effects of exercise and prevent social avoidance produced by inescapable stress in sedentary rats. We hypothesize that increasing D1 signaling will protect against the behavioral effects of inescapable stress, similar to the effects of wheel running. So how we do this is we will perform a cannula surgery and pharmacologically increase D1 signaling by microinjecting a D1 agonist into the dorsolateral striatum 10 minutes prior to inescapable stress in adult male Sprague-Dawley rats. And the inescapable stress in this situation will be an inescapable tail shock, and then behavioral testing will be performed, and another D1 agonist injection will be performed prior to perfusing the rats, and finally PS6 immunohistochemistry will be performed to help quantify the D1 receptors within the dorsolateral striatum. And for some background data, it has been seen that in rats that were stressed, in running rats, the expressing D1 cells were increased in the running stressed rats compared to the sedentary stressed rats. And for our experiment, we seen that there is a trend for inescapable stress to reduce juvenile social exploration, and this is prevented by a D1 agonist being injected into the DLS. So we see that the rats that received saline, our vehicle, have a reduced amount of social exploration compared to our rats that received a D1 agonist who were stressed. And then when we look over to our locomotor activity data, we see that the DLS, the injection of the D1 agonist into the DLS had no effect on locomotor activity. So we can conclude from this that there appears to be a trend for stress to reduce social exploration in vehicle rats, as seen here, but not in rats that received the D1 agonist. And these trends in the data may become more significant after further analysis and exclusion of rats that have misplaced cannula. And any protective effect that the D1 agonist is having against stress-induced anxiety is not caused by rats locomoting more, since it was seen here that the D1 agonist does not increase locomotor activity. And the trend in the data is consistent with the hypothesis that increasing D1 signaling in the dorsolateral striatum during stress could be stress protective. Further studies should therefore determine if increasing D1 signaling in the DLS constrains the dorsal raphe nucleus activity during stress in a manner similar to exercise. And these are my references, and I would like to thank the Mark U Star program for funding this research, as well as the Eureka grant for funding my research. If you have any further questions, please reach out to me. My email address is lorena.alvarez@ucdenver.edu, and I'd love to answer any questions that you have. Thank you.